Welcome, everyone, and thank you for joining us at today's seminar, Accurate Clinical Concentration, Response Predictions for Cardiac Arrhythmia Using a Population-Based In Vitro In Silico Model, presented by Dr. Weihsueh A, Professor at the Veterinary Integrative Biosciences at the College of Veterinary Medicine and Biomedical Sciences at Texas A&M University. My name is Susie Valdez, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I want to remind everyone that today's event is interactive. We encourage you to participate by submitting as many questions as you want during our presentation. Simply click the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have any trouble seeing or hearing this presentation, just click on that Ask a Question box and let us know you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Without further ado, please join me now in welcoming our presenter, Dr. Chiu. I will now turn the presentation over to him. Welcome, sir.
Thank you very much, Susie, for that kind introduction. I'm very happy today to present this webinar about how we're using an in silico in vitro model to predict concentration response relationships for cardiac arrhythmias. I'm giving this presentation on behalf of the Texas A&M North Carolina State University EPA STAR Center. I'll begin with the acknowledgements. This center is a multi-institution collaboration from Texas A&M and North Carolina State University with additional collaborations with Molecular Devices, the National Toxicology Program, ICF International, Cellular Dynamics, and Protein Fluidics. And the funding sources are listed over on the right. The learning objectives today are first to describe some of the current practices in cardiotoxicity assessment for xenobiotics, and then to explain how induced pluripotent stem cell-derived cardiomyocytes may be useful in advancing the assessment of cardiotoxicity for xenobiotics. So first, by giving a brief background as to the cardiotoxicity of xenobiotics and how they're assessed. Then I'll describe an in vitro in silico model that we have developed as part of our center to make clinically relevant predictions for an arrhythmia called QT prolongation. And then I'll end with some implications and future directions for this research. So cardiac toxicity is a known hazard for pharmaceuticals. A large fraction of drugs that have been pulled off the market or that never make it to market are due to cardiotoxic events. And in fact, for post-approval adverse event reports, cardiac arrhythmias are the leading number of those types of events. So the question then, based on our experience with pharmaceuticals, is whether this also might apply to, say, environmental and industrial chemicals. So for air pollution, this is known to be an issue as well, both for cardiovascular disease as well as other types of biomarkers like QT interval. They appear to be affected adversely by air pollution. Other exposures, the question is more open. There's little data beyond some epidemiologic studies on a few chemicals that are very well studied. In addition to air pollution, lead, environmental tobacco smoke are other examples, and arsenic. But because it's not routinely tested in experimental animal studies, and I'll get into that in a little bit in a few slides, it's really an open question as to the broad swath of the tens of thousands of chemicals that are in commerce, whether they might induce cardiotoxicity in humans. In our work, we are using QT interval as a biomarker of cardiovascular risk. This is because it is a well-established biomarker for both genetic and drug-induced cardiotoxicity. and is known to increase the risk of sudden cardiac death. For congenital QT prolongation, those individuals, once they're identified, are prescribed pharmaceuticals in order to blunt their heart rate, so as to prevent arrhythmias from resulting from the QT prolongation. And drug-induced QT prolongation is a major issue for instance, chemotherapeutic drugs, because those individuals can't avoid using those drugs, but the adverse effects of cardiotoxicity can be an issue during treatment. In addition, there's an emerging literature over the last 3 to 5 years that even baseline QT interval is a risk factor in the general population. And a number of observational studies have associated QT interval in the normal range with increased incidence of cardiac death, major cardiovascular events or death, and other effects such as stroke and atrial fibrillation. The molecular and physiological basis of these risks is thought to be as follows, and is shown on the right side here. With the QT prolongation being prolonged, this is due to a delayed action potential in the heart. This delayed action potential then leads to an increased likelihood of arrhythmias, particularly one called torsade de pointe. And this arrhythmia then this very severe arrhythmia can then increase the likelihood of myocardial infarctions, which then, of course, increase the likelihood of death. And so it's presumed then that in the population, if there is an increase in the QT interval across the population, that'll then increase the incidence of adverse cardiovascular events in the general population. Because of these concerns, the first, drugs are now routinely tested for potential cardiotoxicity. During early drug development is a combination of in vitro and in vivo assays, which provide evidence or lack of evidence of cardiotoxic risks. But in late drug development, during phase 1 and onwards, even if there are no preclinical concerns, there is still the requirement for what's called a thorough QT study, the TQT study. And this is a multi-million-dollar clinical trial that intensively measures the QT interval by EKGs in volunteers that are exposed, that are administered these drugs. I should emphasize that the reason why this trial is required, even without preclinical concerns, is that those assays in early drug development are not always sensitive enough to detect potential concerns once it goes in vivo. Now, the threshold of regulatory concern that FDA and the international regulatory bodies have established is that the upper 95% confidence bound for the change in the QT interval cannot be more than 10 milliseconds. And since this has been implemented in the last 10 or 15 years, this has been highly successful in reducing cardiotoxicity in approved drugs. On the other hand, for other types of xenobiotics, there really is no safety testing strategy for cardiotoxicity. Even for compounds that are intensely tested in preclinical models, such as pesticides, there is no general approach to addressing this effect. And some of the reasons are shown here. First is that the rodents that are typically used, even in those in vivo studies, are fed low-fat diets and are not monitored for arrhythmias and cardiotoxicity beyond gross pathology or histopathology. And effects on arrhythmic effects do not often show up in pathological examinations. Second, the main preclinical models that do seem to be somewhat predictive of cardiotoxicity, such as dog models, are not routinely used for non-pharmaceuticals because they are more expensive and they have a smaller end that you can afford for in conducting those safety tests. So that ends up meaning that the cardiovascular effects of chemicals in the environment and in industry is mostly based on epidemiologic data, which means that it's not really safety testing. This is assessing effects that are already occurring in the population. So some of the limitations of the current approaches are, first, the drug testing world, the preclinical models and even the thorough QT study is thought to have high sensitivity, but there are some questions raised as to its specificity. There are also high cost, whether it's dog studies or the clinical trials, they are much higher cost than preclinical studies. Third, there's the issue of population variability. Even the regulatory threshold by FDA is a mean effect and doesn't necessarily characterize susceptible subpopulations. And then fourth, you can't conduct clinical trials on non-pharmaceuticals because of ethical concerns. Now, FDA, of course, is aware of this, and in collaboration with industry is addressing some of these issues, particularly the first two, through its SIPA initiative, the Comprehensive in Vitro Pro-Arrhythmic Assay initiative. Our center at Texas A&M and North Carolina State University is addressing these other challenges and also in the context of non-pharmaceuticals. The basis of a large part of our work is to take advantage of the potential for induced pluripotent stem cell-derived cardiomyocytes. These are fairly low cost, especially compared to clinical trials. They're actually commercially available through companies such as Cellular Dynamics, which is a Fuji company. There is the potential, in addition, for population-level and patient-specific screening. It's not just one cardiomyocyte line that is available, but there's multiple lines derived from multiple individuals. And there's been interest in the precision medicine world of using patient-specific induced pluripotent stem cell-derived cardiomyocytes to do precision medicine. Now, these cells, once they have been coaxed into becoming cardiomyocytes, beat synchronously in vitro, and they exhibit a lot of the types of responses that are expected from congenital as well as drug-induced cardiotoxicity. And because they are derived from induced pluripotent stem cells, and they are also commercially available, these are reproducible resource. And unlike primary cells, which are different each time. So about a year and a half ago, we published a proof of principle in which we looked at drugs, pesticides, and some other industrial chemicals in a cardiomyocyte model. And we found that indeed, first, as expected from other previous literature, drugs that are known to cause QT prolongation, say, will prolong QT, or the analogous arrhythmic pattern in vitro in this system. But in addition, it was found that a number of pesticides, as well as PAHs, polycyclic aromatic hydrocarbons, also seem to affect cardiac rhythm, whether it's increasing or decreasing the beat rate, or inducing something analogous to arrhythmias like QT prolongation. So this seems to suggest that indeed cardiotoxicity hazard might exist in environmental chemicals, given that these are known to work for congenital and for drug-induced adverse effects. And it appears that some environmental chemicals are also causing these effects. So we wanted to take this further from not only demonstrating that it can occur, but trying to actually quantify the concentration response relationship so that regulatory decision-making can be made on this endpoint for xenobiotics that are not pharmaceuticals. So our central hypothesis is that a population-based in vitro model combined with in silico pharmacodynamic modeling can lead to an accurate prediction of in vivo results equivalent to an in vivo clinical thorough QT study. So in this study, even though our ultimate goal is to address environmental chemicals, we're focusing here on drugs for which there is clinical thorough QT type data. So we're trying to predict, first, the concentration response relationship in terms of the actual change in QT as a function of concentration. And then second, whether we can actually quantify the range of concentrations that would satisfy a particular regulatory threshold, such as that for drugs that's promoted by FDA. So with that, I'll go into now some, what our model is and how we developed it, as well as the results. So our study design, we took induced pluripotent stem cells that were available for 27 different individuals. And then we looked at its effects on 13 different drugs, 10 that were positive for in vivo QT prolongation, and 3 that were known to be negative and have been thoroughly tested in clinical trials for QT prolongation. So then we have, this is examples of different beat patterns for the top row is at controls for just DMSO, and then and media, and for 3 different representative donors. And as the concentration increases going down, you have changes in the beat pattern, and at higher concentrations, you see that characteristic arrhythmia in which the beat doesn't fully decline to baseline and plateaus for some time before going down to baseline. And that is sort of the in vitro equivalent to a QT prolongation. So these beat patterns were then quantified using a peak processing algorithm. Then we use Bayesian population modeling to model the concentration response relationship. And then as well, we use rapid equilibrium dialysis to relate the concentrations in our assays to concentrations in vivo in order to have a full prediction as to the in vivo concentration response relationship for QT prolongation. And then those were then compared with literature concentration response results. So just a little bit more detail about how this comparison between in vivo and in vitro works. So qualitatively, on the in vivo side here, there's a normal EKG, and then the QT prolongation is the interval from the Q wave to the T wave, and how that increases with increasing concentration. And then with Torsade, you get this very irregular, very rapid beating pattern before you get to quiescence when the heart is no longer able to beat. Now in vitro, you have normal beating, which doesn't have the same amount of sort of detailed wave patterns as the EKG does. But when you get to QT prolongation, you get this characteristic notch formation in which the peak rises at a normal rate, but then then there's a delay in how it's able to decay down to baseline. And then at higher concentrations, you get the very rapid small beating analogous to Torsade de Pointe, and then eventually it gets to quiescence at even higher concentrations. So quantitatively, we used published pharmacodynamic modeling results for concentration response relationships for QT as sort of our gold standard. And then we modeled the change in this decay rise ratio as a function of concentration from our in vitro system, and then we simply compared them. So this comparison then requires a number of different conversions or commonalities. First is what is the dose equivalence? And so here we presume that the free concentration, that is the concentration that's not bound to protein, should be the same when compared across in vivo to in vitro. So for in vivo, we use literature-based values for what the free fraction in serum was to scale the total concentration from literature to free concentration. And then in vitro, we actually measured using rapid equilibrium dialysis what the free fraction in serum and cardiomyocyte media was to convert those concentrations that were measured in vitro or applied in vitro to a common metric. And then the other question is, what is the common effect metric? So the beating of in vitro cardiomyocytes is somewhat slower than typical, normal in vivo heart rates because there's no pacing. These are just spontaneous. This is the spontaneous beat rate of the cardiomyocytes. And so we, based on other toxicological principles, we posited that if you change both of these to a percent change, so a 5% change in QT prolongation from baseline and a 5% change in the decay rise ratio from baseline, that this would produce a common effect metric that you could then compare, given that the baseline beat rates are so different between in vivo and in vitro. And then, of course, we only restricted the comparisons to the Cmax that's achieved in vivo, since that's the range over which there's actually in vivo data. So in terms of clinical translation, we then took these concentration response relationships and then used baseline QT QT values in the relevant in vivo population, then converted the concentrations back to plasma concentrations, and then predicted that plasma concentration of each compound that would be predicted to have higher or lower than a 10 millisecond change in QT. And that then is our sort of thorough QT study in a dish, where we basically are modeling an entire clinical trial in an in vitro system. So then what are the results? So this is an example with disopyramide of the concentration response relationship for the decay rise ratio across 27 different donors. And as you can see, in each case, at higher concentrations, there's an increase in this decay rise ratio that is characteristic of QT prolongation or delayed action potential, and that the model is able to fit each of those very well within the range that there's data available. So all 10 of the positive control drugs exhibited this increase in decay rise ratio across multiple donors. And as well, at the highest concentrations, there's often the notch formation that I mentioned previously. For the 3 negative control drugs, some of the donors did actually exhibit an increase in the decay rise ratio, but none of them had that notch formation indicative of a more severe arrhythmia. So our first conclusion then, with respect to hazard, we do seem to are able to predict that QT prolongation will occur for the positive drugs. The question then is, quantitatively, do we have that, is the relationship actually accurate quantitatively? Now, I should note that even for this sort of qualitative results, the known positive compounds had predicted effects when you look only within the range of up to the Cmax of between 1 and 46% at the Cmax, whereas the known negative compounds, if you only go up to Cmax, the predicted effects were less than 1%, usually much less than 1%. This suggests that the negative compounds for which we saw increases in the decay rise ratio, those were concentrations that were beyond the clinical doses. And so it's within the range in which people are actually exposed, they seem to be consistent. So quantitatively, we can also make this comparison, graphically here, where in each case here on the left, for disopyramide, this is the predicted concentration response relationship for, as a function of free concentration, the percent change, compared in vitro, which is the colored shaded regions for the confidence intervals, and then in vivo, which are the purple and black lines. And as you can see, they lie right on top of each other, which is really a stunning result, that with no additional sort of fudge factors or any type of calibration, just using from first principles, free concentration, percent change, and this cardiomyocyte model, we're able to have very accurate prediction of the in vivo response. Similarly, on the right, for a negative control, again, for the in vivo black line, it only goes up to the Cmax. So within that range, even though at the highest concentration there is some effect in vitro, within the range in which is relevant clinically, there is no effect, and the two results are completely consistent. So looking then across all the different compounds, over on the left here is the population median across our 27 individuals for each of the compounds for which there is a positive, is a known positive. And as you can see, the gray line diagonal is exactly equal, and then the dotted lines are threefold up above or below. Almost all of our positive controls are within a threefold of the in vivo result in terms of this is the concentration at which there's a 1% change in QT. Now, one of the reasons why we used a population of 27 individuals, which is actually coincidentally larger than some of the clinical trial populations, is that we were concerned that if we just use a single donor, that donor might be idiosyncratically either more sensitive or more resistant to these effects. And indeed, the quote-unquote standard donor that's sold by CDI seems to be more sensitive than the population median. So if we had only used a single cell line, we would not have had such such accurate results. As you can see here, not only is the scatter wider, but also it tends to be biased towards a higher, a more susceptible effect. So then in terms of clinical translation, this involves determining the range of concentrations for which there is a concern that is greater than 10 milliseconds change in QT. And this is shown on the graph on the left here. And so the ranges that are below the threshold of concern are shown by the shaded regions at the bottom. And then the probability that the change in QT is greater than 10 milliseconds is shown by the curved lines. And then the Cmaxes are shown with the inverted triangle. And so as you can see, all the positive controls, except for moxifloxacin, clearly fail the regulatory safety threshold at the Cmax. And that is, of course, as is expected. That is, that inverted triangle is above those shaded regions that are at the bottom of each graph. And then the three negative controls, two of them, but except for lamotrigine, clearly satisfy the regulatory threshold. Now, interestingly, for the two that are more ambiguous, moxifloxacin and lamotrigine, these have different conclusions at the population level versus at the individual level, meaning, for any individual random donor, it might not satisfy the regulatory threshold, even if for the population median it does. And this is actually consistent with the clinical literature in which these two compounds are a little bit more ambiguous as to whether they are quote-unquote safe or not for this endpoint. So it appears that our central hypothesis is supported, that we can predict actually the concentration response relationship, and we can predict the range of clinical concentrations that satisfy the typical regulatory threshold. So I'm going to end now with some discussion of some implications in future directions. So first is that this successful model really had 6 key components that were all needed in order for it to make these accurate predictions. First is that it used a population of induced pluripotent stem cells rather than a single donor. And as was shown in the earlier scatter plot, the single donor might be inaccurate in terms of comparing to in vivo concentration response relationships, but taking the power of the population, you average out the individual idiosyncrasies of individual people. Second is that the decay-rise ratio appears to be an accurate surrogate for the in vivo QT. And that was not necessarily known to be the case. A lot of previous literature had really focused on that notch formation rather than a continuous measure such as that ratio between the decay time and the rise time. Third is that the Bayesian population modeling of concentration response. It's essentially a random effects model in which there is a shrinkage, statistical shrinkage by using multiple donors, allowing for a more confident estimate of the population central tendency. Fourth is the use of free concentration and percent change to make that equivalence between the in vivo and in vitro concentration response relationships, since they are in different units using different media versus serum, and given the difference in baseline beat rates with between in vivo and in vitro. Fifth is the development of a clinical translation model that then can actually evaluate whether a compound satisfies or does not satisfy a particular regulatory safety threshold. And then sixth is something I haven't talked about much, but this experimental protocol is suitable for high throughput screening. We use 384 well plates in our screening using calcium dye to measure the calcium flux. And this is much less onerous than some of the other types of cardiotoxicity in vitro systems, which involve actual electrical measurements. Of course, there are a number of limitations. We only tested 13 drugs, but this really was limited by the availability of published thorough QT clinical data that included the pharmacokinetic pharmacodynamic modeling so that we could extract the concentration response relationships. Second, it doesn't address some of the more severe indicators of cardiotoxicity. These are really early biomarkers. The QT is an early biomarker of potential risk. And so for those who are interested in more severe effects, we have not yet extended the model to those. And then third, with 27 individuals, we can't really describe accurately the range of population variability, as in, whether there's an idiosyncratic population. There would need to be a larger population to be assessed in this model for that to be the case. But the high throughput screening format using 384 well plates would enable this type of work to be done. The vision then is a completely predictive in vitro-based model for chemically induced cardiac arrhythmias. So starting with ADME, there's a lot of work from SimCip and such using in vitro and in silico models for pharmacokinetics, and then the middle part using our in vitro model and in vitro and silico model for the pharmacodynamic portion to get to QT prolongation. And then at the tail end, there's a number of published cardiovascular risk models that have started to incorporate EKG measures such as QT in their risk prediction. And in some cases, these models claim to have better prediction by including QT and other types of EKG markers. And so by connecting these different models together, we would have a seamless handoff from exposure all the way through population changes in population risk for cardiac arrhythmias. So the key messages I'd like you to take away are these 5 bullets here. First is that thorough QT studies are known to be highly resource intensive, even though they are currently required for regulatory safety assessment of drugs. We asked here whether we could use an in vitro population-based model combined with in silico modeling to accurately predict what these thorough QT studies, these clinical trials actually measure. And also to go beyond just sort of academic interest, but to actually have an output that could be used in a regulatory setting as to the presence or absence of regulatory safety concerns. We found that for 10 positive and 3 negative control drugs, we were able to accurately predict in vivo concentration QT relationships, as well as accurately predict the presence or absence of regulatory concern. And so for pharmaceuticals, this thorough QT in a dish type model could potentially reduce the need for clinical thorough QT studies in the future. And then I think one of the large unknowns here is for non-pharmaceuticals, since there's so little known about the potential cardiotoxicity of environmental chemicals. And here, the high throughput format of this thorough QT study in a dish can really fill a critical gap in terms of understanding both the hazard and concentration response relationship for chemicals to which we're all being exposed. And with that, I thank you for your time and attention.
And thank you, Dr. Chiu, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to remind everyone that today's webcast will be available for on-demand viewing through May 2019. As a final reminder, Dr. Chiu will follow up with any questions you submitted via email. That's all for now, and thanks for joining us. We'll see you next time. Bye-bye.